Hello, everyone, and welcome to the last part of the adaptive immune system. And where we're really going to be talking about a different type of an adaptive cell, the B cell. This most notorious cell that produces these Y-shaped proteins that we call antibodies that protect us. And I'm quite sure you've definitely heard about antibodies, specifically given the pandemic, and where we've actually had a heavy we've been relying on them quite heavily for them to protect us against infections. So, what is the goal of a B cell development? Well, what you're trying to do is you're trying to create B cells that have actually a receptor that actually are able to, determine or let's say kind of bind to a huge range of antigens. So these receptors have to have a huge diversity. Well, similar to your T cells, we get these actually diversity by combinatorial diversity. Just like your T cell, they have the VDJ segments. Well, your B cell also has it. So you actually get different combinations of VDJ segments. Hence, you have a combinatorial diversity because you're using different V segments, different D or J segments, putting them together, creating different, let's say, B cell receptors. But when we create, let's say, a segment, we actually have this addition of these non-templated nucleotides, which we call them the N nucleotides. These are just random nucleotides adding there. That also gives us a high diversity. We get different types of receptors because of that. But one thing that also B cells have extra compared to T cells, they have something called somatic hypermutation, which is literally adding random mutations to the VDJ segments. And we'll be talking about it a bit in today's chapter. But of course, we want to create a B cell that has a certain receptor that's only able to bind to pathogens and not to self proteins. Because if you have self against self proteins, autoinflammatory diseases. And we're going to talk a lot about that later on next week. But for now, I just wanted to talk about how does a B cell actually obtain its receptor. So, as we said, B cells are part of the adaptive immune response, which is basically composed of your T and B cells. Different to your innate immune response actually is able to, let's say, bind or able to actually get activated by looking at different recognition, basically, let's say, patterns within viruses or bacteria. So it's not so specific because you're looking at patterns. When it comes to B and T cells, they're looking at specific, extremely specific antigens. And they're able to recognize them. So hence, these are much more specific. So these antigens are, as we know, any molecule or part of the molecule that is recognized by your basically T and B cells. And they are able to kind of cause, let's say, an immune response. And generally, antigens are proteins. They can be glycoproteins in bacteria, but most of the time, they're basically proteins because they're able to bind very well to the MHC. When it comes to the B cell, as we know, B and T cells, they all develop in your, they all basically are created in your bone marrow. T cells are more, let's say, developing in your thymus, hence why the T. B cells, their primary development occurs in the bone marrow, hence the B in B cells, so bone marrow. Later on, once they're now basically kind of mature at the beginning of the stage, they leave actually the bone marrow and then they go to lymphoid organs. This could be the spleen or this could be lymph nodes, where they will basically finalize their maturation step. And once they basically are mature, their biggest role that once they're mature and once they're activated is of course to bind through their B cell receptor. They bind to specific antigens and they're able to get activated and produce antibodies. They produce antibodies against extracellular pathogens because antibodies are not able to cross the cell membrane. They do not actually are able to cross hydrophobic regions, so they're mostly binding to extracellular basically pathogens, be it bacteria, be it viruses that are on the outside, not on the inside. You do not use them on the inside, or parasites or other stuff that are found in the extracellular. And in development, the B cells is usually basically can be antigen dependent at the beginning, and we'll be talking about this a bit later. But of course, B cell development, just like in your T cell development, it's very guided by a lot of cytokines. For example, in the B cells, you can use interleukin 7 that helps B cell lineage development, interleukin 4 that kind of promotes survival or maturation, or then you have 21, which really pushes B cells to become plasma cells, which are basically a subtype of B cells that are able to produce antibodies. So I don't need you to remember these cytokines, hence why they're literally in red. But just want you to know that, this maturation is very directed or heavily directed by interleukins or these kind of cytokines like 7, 4, and 21. So if you look at basically your B cell receptor, so this receptor that's able to recognize specific antigens, it's quite similar to your T cell receptor, but in the B cell receptor, it's literally made of an antibody. So knowing the structure of an antibody is literally like knowing your B cell receptor, because it's literally an antibody that's stuck on the membrane of your B cell, and that becomes your B cell receptor. So the B cell receptor, like your T cell receptor, is made of two different types of chains. The B cell receptor is made of your heavy chain, which gets its name for being the bigger chain, so hence heavy. And there's two heavy chains that are connected to each other through disulfide bridges. Then you have two other basically light chains, and the name comes to light because they're the smaller chains that are bound to the heavy chains, and they're bound through a disulfide bridge. So they create this really nice Y-shaped kind of protein. So similar to what you have in your T cell receptor, you have these kind of domains. You have the variable domain and you have the constant domain. So the heavy chain basically has, as we said, a variable domain and a constant domain, shown here. So the VH basically means it's the variable domain of the heavy chain. The CH is the constant domain of the heavy chain. And just like you have with heavy chains, you also have with the light chains, you have a variable domain and a constant domain. So again, similar to your TCRs, the variable domains are the ones that are basically changing. They keep changing, and they're the ones that are literally binding to your specific antigens. And this is where you need to have a high diversity, because you're binding to millions of different types of antigens. You need to make this extremely variable and diverse to do that. So hence, the variable regions that are found both on the heavy chain and the light chain are the ones that are binding to your antigen. The common chains, the heavy, basically the common, the constant region of the heavy chain and the light chain, they basically are quite, constant per se. When it comes actually to your light chain, you only have two different types. Actually, you have either kappa or lambda, and they're actually almost the same. They have the similar, let's say, function. So they're just more there for structure. When it comes to the heavy chain, its constant regions, there's basically in humans 5 different types. There's the IgA, D, M, or E, or G. So basically they have different constant regions. Either it's an IgA, D, M, E, and G, and they can actually give you different functions. For example, if you have an IgE, this antibody is more for allergic reactions. If you, for example, have an IgG, it's more for bacteria. If you have an IgA, that is more actually for mucosal sites. If you have IgM, it's much more for complement activation, actually. And we're going to talk a lot about them in generally. So, hence, the constant regions, they're constant because they're usually within one antibody, they're the same. So if you have IgG antibodies, the IgG heavy chain is all the same. What differs is, of course, between antibody and antibody that are IgG, there is the variable regions, hence the name comes from there. So it's the constant heavy chain that, determines the isotype. Either it's IgA, D, M, E, or G. I will talk about all the different types of them that exist. So let's, dissect this into smaller parts. So let's just take now the heavy chain. As I said, the heavy chain, it literally corresponds. It has two regions, the constant region, and then you have basically your variable region. So in your variable region, that's where you have the CDRs. And if you remember, the CDRs are basically your complementary determining regions. These are the ones that are actually being changed constantly, and they're the ones that they're binding to your antigen. So literally, those are the regions that are basically being changed. And your variable actually region or domain is actually made of VDJ segments. Similar to your beta chain, your TCR, we have these VDJ segments, and where we have many different types of VDJ segments, which we can actually create different combinations. This is what gives variability towards that domain. Then in your heavy chain, in your constant region of your heavy chain, this is where we have basically the 5 different types. We have the IgM, the IgG, A, D, and E. So it depends on the chain type. So if we have a micro constant heavy chain, that gives you IgGM. If you have a gamma, gives you IgG, alpha IgA, delta D, epsilon E. So we use this mnemonics in a sense where the Greek alphabet denotes the basically kind of English alphabet. So if someone tells you what determines the isotype of the antibody, it's the constant region of your heavy chain, because they can be of the IgGM, IgG, A, D, and E. And they really can tell you a lot about an antibody. We will be talking about that later. So your light chain. So basically you have 2 different types of light chains. You have the kappa or lambda, basically, which basically will have these different they're basically different type of domains. So what you have basically, of course, in your light chain, we know that there is a variable domain, so hence one V domain and one basically constant domain. So the constant domain stays the same, so it's either kappa or lambda, and within the variable region, you have your different three CDRs. So the CDRs are made of basically the VJ segments. So this could be your alpha TCR chain. So because the alpha has VJ segments, well, the same thing goes with your light chain. So your variable region in your light chain has 3 CDRs. So you have 2 light chains. So for the light chains have 6 CDRs in total. When it comes to your variable heavy regions, you also have 3 CDRs in each. So you have basically 6 for your heavy chains. So in total, you have actually 12 CDRs. Basically 6 coming from your heavy chains and 6 coming from your light chains. And similar to what we see with the TCR, we try to actually create different types of combinations, because as we know, there's multiple different V chains, multiple different H chains, and basically or J chains. And what we can do is we can actually create multiple different combinations. For example, we can have, in the light chain, we can have V20 and J11, which is different to V1 and J3, for example. Or in the heavy chain, we actually have V2, D13, J29. So we just try to create different combinations, and this gives us a level of diversity. So let's now talk about how do we actually create a B cell. So as we know, all your immune cells, they start off from your hematopoietic stem cell. Your hematopoietic stem cell basically starts to create a lymphoid myeloid primed multipotent progenitor, which basically means this can still become either myeloid or lymphoid. And if it's going to your lymphoid, it will start to create a common lymphoid progenitor, the CLIP that we know. This CLIP depends on the environment. It will basically become either a B cell or a T cell or an NK or a dendritic cell. Depends where you put it. You put this CLIP in your thymus. The thymus has certain cytokines, or let's say molecules, that push the CLIP to become a T cell. But if you put this CLIP in your bone marrow, the bone marrow has the perfect cytokines and chemokines to differentiate it into a B cell. So it depends where the CLIP is. So this is basically something you have to kind of remember. And basically, if the CLIP is in your bone marrow, it starts the primary stage of development, and where your B cells basically start to develop into immature B cells. Once you become an immature B cell, this B cell is able to then leave your bone marrow. It's then able to actually get into your blood, and it's then able to go into peripheral lymphoid organs or secondary organs, like for example the spleen or lymph nodes, and where they actually start to finally fully mature. I will basically go into details about all this. So as I said, you basically start off your stem cell. In this case, is your CLIP, your common lymphoid progenitor. It's basically in your bone marrow. And what happens is that basically, once they're in the bone marrow, they get certain, let's say, cytokines and molecules that tells them, like, okay, you're going to become a B cell. So the first stage that it becomes is called an early pro-B cell. And once it hits the stage, it starts to create the heavy chain. And it does that by doing the DJ rearranging. So they're trying to get basically certain D segments and a J segment to combine together. And basically once they join together, the early pro B cell becomes a late pro B cell, and where now basically you start to get the V rearrangement. So basically now you have your DJ segment. Now you're adding a V segment to it to fully now create your heavy chain. Once you actually create your your basically your heavy chain, once you're done with the whole VDJ rearranging, now you enter the large pre-B cell. So in the large pre-B cell, we know that we have a heavy chain because we've arranged the VDJ. We have the perfect, basically, heavy chain now. What we do, very similar to what you see with T cells, we actually create a pre-B cell receptor, and where we have basically the heavy chain, but we create a surrogate light chain. So this is a pre-light chain that is able to bind to your heavy chain, and you create a pre-B cell receptor. So this is just to kind of put on top and to be able to signal to your large pre-B cells like, okay, now it's time to start proliferating, to start surviving, and to start to create your light chain. So the large pre-B cell enters this small pre-B cell stage, and where you start actually having the VJ rearrangement, which creates your light chain. So once you have the light chain done, now you have the full pre-B cell receptor. Sorry, your full B receptor, because you have the full heavy chain and light chain. And now this can be actually put on your B cell, which then gives you into your immature B cell. So your immature B cell has a full B cell receptor. And it's an IgM because the heavy constant domain is always, it's literally automatically put in the micro chain. So you actually have the basically your micro chain and your heavy chain. So that means basically that the heavy constant domain is micro, meaning that it's IgM. So the first antibody that the B cell creates is always the IgM. So now you have your immature B cell. All of this occurs so from the stem cell to your immature B cell, this all occurs in your bone marrow. Once that is done, the immature B cell is able to leave and then basically go into your blood, which then goes to your secondary lymphoid organs like your spleen or your lymph node to become an immature B cell. And in there, when they become an immature B cell, they actually create another B cell receptor called IgD. So basically, it's like IgM, but the constant domain of the heavy chain is basically of delta. So it's IgD. So how do they rearrange their DNA? As we said, between the light chains, basically, we know the light chains, there's the kappa and the lambda. So basically, you have different variable. So we know that the light chains, they either have the variable regions have a V or a J. They do not have a D. They do not have that. So the variable and the joining basically chains, they have different combinations that we can actually add together to create, let's say, one variable region. The heavy chain has the VDJ. We know that we're trying to rearrange all these segments to create our variable regions. How do they do this? Well, it's exactly the same as your T cell receptor. So basically, and when it comes to, for example, the heavy chain, you start off with a DJ recombination, as we said basically here. And then once you're done with that, you actually add your V segment, and voila, you have your variable region of your heavy chain. And how that's done is because we basically, these VDJ chains, they have a RSS sequence, this recombination signal sequence. They follow this 12-23 rule that we know from the T cells, and where RAG1 and RAG2 are able to bind to them and kind of cut them. So these recombination steps are exactly the same as the T cell receptor, which I'm not going to go into much detail about because we've already spoken about. But let's go put this into more context, this whole damn thing. So we basically start from your early pro B cell. So your early pro B cell does not have no B cell receptor. So there's nothing. Once it's in the early pro B cell, you first have basically your VDJ rearrangements. So when you have your VDJ rearrangements, you finally have your heavy chain. So once you have your heavy chain, what we do is we create a surrogate light chain. So this is your pre-light chain that's going to bind to your heavy chain, and you're going to create your pre-B cell receptor. This pre-B cell receptor is perfect. You can add it on the membrane of a B cell. But the issue with this one is that even if it binds to an antigen, It doesn't have any signaling capacity. It's not able to signal to your cell anything. So what we do with your pre B cell receptor, we add it together with basically these two kind of chains, which are, we call them the Ig beta or Ig alpha, or CD79 alpha or CD79 beta. And these are signaling molecules. They're two little molecules. They bind together with the pre B cell receptor. And what happens is that when the B cell receptor is basically actually binds to something, it kind of signals to these two. It's like, okay, something is happening. We need to start to creating a signaling cascade to the B cell. So this kind of tells your B cell to start to survive, start to proliferate, and start to create your light chain. And that's what happens. So basically, once you start to entering your immature B cell, you create your light chain, which now is able to bind to your already formed heavy chain to create your full antibody. So as we know, the full antibody is of the IgM. So because the micro heavy common chain is micro, so hence IgM. So that's not put in. The IgM antibody does not also have a signaling capacity. It has to be associated with CD79 alpha and beta, because once the IgM is able to bind, it's able to signal these guys to do a signaling cascade in the B cell. So once you have basically the IgM, this is an immature B cell and it's able to finally leave. So it leaves to become a mature B cell. So as we said, one layer of basically them gives in some sort of way high diversity to your B cell receptors is the VJ recombinations. You have so many different recombinations that you can actually do, which gives you different B cell receptors that are able to bind to different things. But we also have this junctional diversity in where when we're basically literally trying to join the V or D or the G or J segments, we basically have the addition of this non-templated nucleotides, which are random, and they literally are joining together these basically segments. And this is random, so you're literally giving much more diversity, creating much more different CDRs that are binding to different proteins or antigens. So once you're basically done, basically here you have your mature B cell, you have your basically your B cell receptor, which is an IgM. This is able then to leave, and it leaves basically the bone marrow, and it basically goes into your blood. We call these transient B cells because they're basically in transit. They're literally just moving around, and they basically enter technically either your spleen or your lymph nodes, and there is where they're gonna start to actually encounter antigens, because now they have actually a B cell receptor that's able to bind to an antigen. So depending on where it binds to the antigen, basically when it binds to the antigen, this can activate it, and it can basically push it from an immature to a mature B cell. So that occurs in your lymphatic tissues, in your secondary lymphatic tissues, be it in your spleen or your lymph node. So let's imagine that it enters your lymph nodes in this case. As we know, basically these will enter your afferent lymphatic vessel. It's going to go inside, and they're literally going to actually go into your primary lymphoid follicles, and there is where they're going to be exposed to antigens. So if a B cell, this immature B cell, is able to bind to an antigen, that kind of signals it like, okay, I need to get activated. And this activation can be just purely antigen based, or sometimes what happens, or majority of the times, is that you have T follicular T cells. So these are T cells that are also there, that they literally will go and bind to your B cell and will actually secrete interleukin 21 and also STAT3. So basically, interleukin 21, which then will basically activate STAT3 on your B cell. This STAT3 is a transcription factor that really pushes the B cell to proliferate, differentiate, and create a follicle. So we call these primary lymphoid follicles, which then form germinal centers. So these germinal centers are literally our centers where B cells are proliferating, and they're basically becoming much more mature. So this is where once they're active, they create these germinal centers, or we also call them secondary lymphoid follicles, where you see B cells are just getting activated, proliferating, and to start to create antibodies. So once these B cells are activated, two things can happen basically. First of all, basically these B cells, they are in the mode that they will only create IgM antibodies because they can only produce the basically micro heavy chain. Sometimes you don't want that. Sometimes you literally want to change from an IgM to an IgG to an IgE to an IgA. It all depends on basically the context. If you have a bacteria, you want IgG. If you have basically an infection in your mucosal sites, you want an IgA. If you want to activate basically your allergic system, you want an IgE. So what happens is that technically your antibodies, they do something called your class switching, and where basically now suddenly this is your IgM, this is now changed. Your constant heavy chain is changed to depend to whatever you want it, to an IgG, to an IgE, whatever. But with also what happens is basically the antigen, these CDRs in your variable regions, both in your heavy chain and your light chain, they also go an additional round of diversification, something that we call somatic hypermutation. So I'll talk about both of these. So when it comes to somatic hypermutation, what happens is that, okay, now we have antibodies that are able to basically, let's say, bind to your antigen. Let's imagine influenza. We want to make sure that we can create an antibody that it has a perfect affinity towards that influenza virus. Well, what happens is that we basically start to actually add massive mutations at a very high rate to the CDRs. So so basically if you go here, so these are basically your variable regions. These literally black bands are indicating these are different mutations that we're adding to your variable regions. And what is that that's causing it? It's actually this kind of enzyme that we call activation-induced cytidine deaminase. So you have this enzyme called AID, which literally deaminases cytidines. So it creates cytidines into you. So you're creating so many different mutations, which now you're further pushing the combination. So now you're basically having so many different types of antibodies in a certain manner that you're trying to create the perfect antibody that's able to bind to your perfect antigen. That's what this is doing. And this is AID is usually induced by interleukin 4. That's usually given by your T follicular cells. They produce interleukin 4, which then basically kind of help the actually induction of AID to start creating these point mutations. So you're trying to create much more diverse antibodies, because it could be that the antibodies that you already have are not able to bind perfectly to the pathogen you're being infected with. So let's say, imagine you have the perfect variable region that's able to bind perfectly to your pathogen. You want to basically change your basically constant heavy chain, because all of them are IgGM at the beginning. We want to change them. So we can change them to basically an IgG, to an IgA, or to an IgE. IgD, you don't create them. You don't need them basically. You can only change them from an IgM to an IgG, A, or E. And within the IgGs, there's different subtypes. You can create an IgG 1, 2, 3, or 4. You can create an IgA 1 or 2. These are different subtypes of IgA. I'm not going to go into details about them, but technically you can change the constant heavy chains, and that depends on what you want. So because of basically this different diverse kind of, let's say, basically processes, we can get a huge amount of diversity in antibodies, because what we're doing is we're doing random recombination of gene segments. So we're basically having different VDJ segments from the heavy and the light chains. So that's one source of diversity. We have these mutations at the joining sites. So basically these are the non-templated nucleotides, which also give much more mutations and diversity. And we also, as we said, you're putting a lot of the somatic hypermutations, which are further pushing basically the basic diversity. So suddenly you can have so many different types of antibodies that are able to bind to so many different types of antigens. But one thing that we see something called clonal selection. So let's imagine you create, basically this is your B cell, this is your immature B cell, and now it's getting activated and it starts to create different B cells. So you have this B cell with basically this kind of green triangle. This has a B cell receptor that's able to bind specifically towards the triangles. You have this kind of now pink star. This only is able to bind to pink stars. This is only able to bind to basically, let's say, yellow squares. So if you're infected with your stars, basically it's only the B cell receptor that is able to bind perfectly. That's the one that is gonna proliferate. So the rest are basically dead. So you get a huge diversity because of all of these basically kind of reasons that I just gave, because you're trying to find the perfect B cell that's able to bind to the antigen that you're basically infected with, and that's called clonal selection, because you're literally selecting one clone to basically be the one producing a one type of antibody that's able to bind perfectly to whatever you're being infected with. The rest that are not binding to it, so this red triangle, this green triangle is not binding to this, it dies, this one also dies. So it's only the ones that are basically able to bind beautifully with a high affinity. So now let's go into B cell activation. So as we said, once we actually have your B cell, your B cell is basically has a B cell receptor, which is literally an antibody that's literally stuck inside your membrane. The B cell receptor doesn't have any signaling capacity. It actually binds, as I've told you, to this Ig alpha and Ig basically beta, which are basically the 70s CD. I think it was 78, if not mistaken, 79. Sorry. So these are the CD79 chains, and these chains basically they have these beautiful motifs. They are called immunoreceptor tyrosine based activation motifs. These are motifs that when this B cell receptor binds to an antigen, this kind of allows this motif to actually be phosphorylated in tyrosine, hence tyrosine based, which then can induce a whole signaling cascade in the B cell, so to proliferate and to survive. So let's now go into the B cell activation. So when it comes to B cell activation, there's two different types. There's the T cell dependent activation, and there's the T cell independent activation. So the T cell dependent activation is literally, the name says it, it's dependent on basically T cells. So what happens is that you have your B cell basically will bind to your antigen through your B cell receptor. This gets internalized. So remember, this gets internalized because B cells are antigen presenting cells, and they basically represent it on its MHC2. So what happens, the B cell will go to your T follicular cells, present it, and will basically kind of tell your T helper cells like, okay, help me, I need to start proliferating. So you basically have this basically very similar to what you see with T cells. B cells put it in the MHC2, present it to T helper cell. You have three signals, one coming from the MHC2, the second coming from this co-stimulation, which we know in T cells is the BH7 1 and 2, binding to CD28 in your dendritic cell, but in this case it's CD40 and CD40 ligand, and also your cytokines. So this pushes your B cell to start basically expanding and getting activated. You also then have your T cell independent activation. So the name basically says it, it's independent of T cells. So the antibodies are able to bind to antigens, which are then able to actually, phosphorylate your CD79 alpha and beta chains, which then they basically create this whole signaling cascade, which are things that I don't want you to actually remember. But what this does is you create a signaling cascade that really pushes B cells to further be activated. And of course, this can be done, of course, just through your B cells, but this can also further occur with your B cell activation, these co-receptors. So you have basically your B cell receptor being activated, but you can also have this co-receptor complex, which is made of CD21, CD19, and CD81. These are all bound together, which can also bind to antigens that are, and this can actually push further more signaling cascade. Again, I don't need you to know the signaling cascade molecules like the Finlin or these ones, but just to know that you basically can also have this co-receptor, which is made of CD21, 19, and 81 to further be activated and push the B cell to further proliferate. So once you actually have basically the activation, either through T cell dependent or T cell independent, you have basically two pathways. Either these basically cells become plasma cells, meaning that they actually become some sort of B cell, which we call plasma, that create antibodies. These are becoming more specialized B cells that create antibodies. Or they create memory B cells. These memories are there and they stay for literally almost a lifetime because they're basically now are specific towards, let's say, imagine you're infected with influenza. So influenza A, in this case, these will stay there. And if you're basically infected again with influenza A, these are basically able to recognize it much more quicker. So they're able to become memory B cells or plasma B cells. But one difference that I want you to really understand is that in the T cell dependent activation and the T cell independent activation, the T cell independent activation, you create very short-lived plasma B cells. This doesn't give you the exact amount of, let's say, signaling cascade and signaling for a B cell to really become a proper plasma cell, and they do not live too long. They only produce IgM. But if in the case of the T cell dependent, they get all the cytokines, all the co-stimulation, all these beautiful signaling cascades that really pushes them to actually become memory B cells and actually long-lived plasma cells that can actually produce not just IgM but different subtypes, IgG or IgA or IgE. So now I want to talk actually about the different types of antibodies you can create. So you can create IgG. So this is the most common type of antibody that you have in your blood. It's basically found mostly in your blood, as I said, and it's extremely good at actually binding to extracellular, basically pathogens, be it viruses, be it bacteria, and they're extremely good at activating your complement, at opsonizing. So as remember, opsonizing is flagging basically a microorganism that this is dangerous, or neutralizing endotoxins. And IgG, there's 4 different subtypes. There's IgG 1, 2, 3, and 4. I'm not going to go into so much more details about these, but basically that there's different subclasses of them. Then you have IgM. IgM is a very interesting antibody because it's literally... Sorry, just had something is itching. So basically, it's literally 5 antibodies connected together. So you literally have 5 antibodies that are connected to each other. These are extremely good at literally binding to antigens. Okay. And I was about to sneeze there. So because basically there are 5 antibodies, they actually have a huge avidity. So avidity is literally the multiple or the combination or the addition of multiple affinities together. So they're really good at binding and creating this high avidity. And they're extremely good at activating your complement system. And usually you see IgM just at the beginning of your basically response, or of your primary antibody response. So they're usually mostly produced at the beginning. Then you have IgA. IgA is extremely good at actually neutralizing pathogens and basically specifically in your mucosal sites because it's able to be really good, be actually produced in your saliva, your tears, in your intestinal, genital, or respiratory tracts, so in your mucosal areas. And IgA can be either a monomer, so basically just as one, or it can come as this beautiful dimer, so 2 antibodies stuck together. And they're very good at crossing the placenta because they can actually go into your breast milk. So they're very good for babies, and specifically when it comes to IgA. IgE is a bit different. This is basically mostly found when it comes to allergic reactions per se. They basically will bind to mast cells, which basically are the main cell for allergies. They also have some sort of important, let's say, kind of function when it comes to parasitic infections. IgD, you don't produce it. It's mostly found, if you go back literally over here, it's mostly found in your mature B cells as a B cell receptor, and you really need it mostly for B cell development. So it's not one antibody that is going to be produced. So once you produce your antibody, how can it actually protect us? Well, there's basically 4 ways. It can actually neutralize toxins, it can opsonize, it can activate your complement, or your effector cells. So toxin neutralization. So we know that there's a lot of basically bacteria, for example, botulism, this kind of bacteria that release this diphtheria toxin. the bacteria per se does not kill us. It's actually the toxin that gets released that can kill us, can cause paralysis. So we can create antibodies that's able to actually neutralize it and not allow it to do anything. Well, same thing goes actually to your virus. So technically, when we actually do a vaccine against COVID, we're trying to create antibodies that are able to bind to the virus and to actually not allow it to penetrate or bind to our human cells. Opsonization, it technically literally means that it's binding to your pathogen and it's literally flagging it. It's flagging it to your immune system to be like, hey, start attacking it because something is wrong. So let's imagine when it comes to IgG, because IgG is extremely good at opsonization, the IgG will bind to it, and the IgG will be able to bind to Fc receptors, and specifically Fc gamma, hence gamma is basically more for the IgG. And these Fc gamma receptors are found all over your macrophages and monocytes, and there's different types, the Fc gamma receptor 1, 2, and 3. I don't need you guys to remember this. I'm going to actually flag these in red, but literally it kind of helps your, let's say, macrophages to identify these antibodies that are bound to your pathogen to tell us like, okay, this needs to be killed. And also, as we know, the IgG antibodies are extremely good at activating your complement, specifically through your classical pathway. So, of course, your antibodies and the complement work in synergy because, as we know, once you have basically this literally whole, let's say in this case bacteria, when you have the antibodies binding to it, and you have these basically complement, they can literally opsonize. So both of these opsonize, and these basically bind to the receptors on your macrophage, either to your complement receptors, the C3 receptor, or the Fc gamma receptors, which really help macrophages identify this as something to be eaten. So of course, other than that, antibodies are able to basically bind. So let's imagine here you have, let's say, your target cell. You have antibodies that are binding. These antibodies are able to bind to these Fc gamma receptors. So if this is an IgG antibody, it binds to the Fc gamma receptor. And this can be found also on NK cells. And what happens is that the NK cells, once they bind basically to the antibody, they get activated and they release granzymes. So they actually allows NK cells to detect target cells to kill them. So this could be a cancer cell. So NK cells actually release these perforins and granzymes. And this whole thing is called antibody-dependent cell-mediated cytotoxicity, or in short, ADCC. So this is another basically nice way of, for example, the IgE. IgE is able to really bind to, for example, parasites. It's able to bind to them, and it's actually able to kind of help mast cells to be like, hey, this is, for example, a parasite. You need to go and kill it. So it actually will release a lot of different certain, let's say, cytokines and chemokines, all these different things to actually cause a lot of other cells to come in to the site of infection and kill the parasite. So this is where I'm going to end it.